Hi, my name is Anthony. I'm a graduate student at the University of Cincinnati, and my project focuses on understanding the dynamic responses of lung epithelial cells to influenza A infection and the effect of genotype or genetic variability in humans. To do so, our collaborators infected human upper airway epithelial cells from multiple donors with influenza A or treated with interferon beta and performed single-cell RNA and single-cell attack sequencing at different time points during early infection. We then inferred gene regulatory networks to dynamically model the transcriptional control of the different epithelial cell populations. We used the inferolator to model gene regulatory networks, and the inferolator models gene expression as a linear combination of transcription factor activities. These transcription factor activities describe the extent of which a transcription factor regulates a target gene, which generally is advantageous to using transcription factor mRNA measurements as proxy for TF activities. We were able to identify major cell populations in the upper airway epithelium, such as basal, ciliated, and secretory cells, as well as some of the more rare cell populations, such as ionocytes and neuroendocrine cells. The results in this poster, however, are limited to the major cell populations. We then used the K-medioid classifier that classifies IAV-treated cells as infected or uninfected based on the expression of IAV transcripts in each cell. This allows us to better model infection response and understand how infected and bystander cells communicate with each other. The first heat map shows the normalized pseudobulk gene expression, where each column represents an individual donor. As expected, we observe a cluster of interferon-stimulated genes that we see in both interferon and IAV-treated conditions. But surprisingly, there are striking differences in bystander cell responses, which indicate that infection leads to cell type-specific responses. We can then use differential gene expression analysis to identify gene signatures that define each cell type and condition. Similarly, we have pseudobulk chromatin accessibility showing cell type specific peaks and some IGV shots showing differential accessibility of some cell markers such as FOXJ1 for ciliated cells, keratin 15 for basal cells, and mucin 5A for secretory cells, as well as dynamic changes in chromatin accessibility of ISG15 in viral treated samples. After inferring gene regulatory networks, we end up with a massive hairball of transcription factor gene interactions that we can then analyze in different ways. For example, we performed TF-TF module analysis where we look at the extent of overlap between different transcription factor targets to predict which TFs may be working together to regulate those target genes. And then we can look at the transcription activity matrix to find the conditions in which those TF modules are active in. For example, IRF7, STAT1, STAT2, and IRF9 are predicted to work together, and then we can see that they are active in interferon-stimulated conditions as well as IAV-infected conditions. Now, another analysis that we can do is core analysis, where we identify transcription factors whose predicted targets are enriched for the gene signatures that we defined earlier. Here we can see a sample of the core networks that define each cell type prior to any stimulation at t equals 0. We can then extend the core analysis across time points to identify the time-dependent changes in gene regulation as well as infection-specific gene regulation. For example, in infected secretory cells, ELF1 is predicted to activate HLA class 1 and class 2 genes, which are responsible for antigen presentation in infected cells. We also see IRF1 being active in infected cells, but not in bystander cells, also as expected. Now, alternatively, we can perform CellChat, which is a bioinformatic tool that predicts cell communication networks based on the expression of ligands and receptors in different cell groups. Outgoing signals correspond to the cell population that express the ligands, and incoming signals represent the cell populations that express the receptors of each of the pathways. And each column represents a different donor. We can extend those predicted pathways and intersect them with our core networks to identify the core network that regulates those pathways in different cell populations. Here we see a sample of a core network that regulates a few of the predicted pathways in infected cells, and we can see some highlighted pathways that are either turned on in infected cells or turned off in infected cells relative to their bystander counterparts.